I thought the abstract was excellent. I would encourage everybody to take a look at it, especially the young scholars over here. I mean, that's how you should write an abstract, I think. But today's session is about application of OR and management science methodologies in biomanufacturing. And perfectly for what the seminar series is intended to do, this is about the group's vision and future research directions. Just 3 sentences in terms of where the, to introduce the talk. One is we need to keep in mind, as the abstract says, that the process of making medicines in biomanufacturing is fundamentally different than the production process of conventional drugs. And then the second point over here is there are limited number of papers, few written accounts of success on ORMS applications in biomanufacturing. This group, or pretty much the entire group or large parts of this group, was involved in the Edelman presentation. Strongly encourage you to watch that presentation where they talked about their applications. But here they're going to be talking about the vision and the future directions. Of course, they'll make some reference to their work. Quick introduction on the speakers that we have. Thank you guys again for making the time and for sharing your knowledge. Oscar Repping is an executive director with global responsibility in the development and maintenance of production processes and analytical tools for vaccines and biotherapeutics. He was especially important a few years ago, I think, anyway. But Marc Baaijens, associate director, global responsibility in bioprocess engineering and manufacturing data science at MSD Animal Health. Bram van Ravenstein is Associate Director, Operations Lead in Bio and Bacteriological Production at MSD Animal Health. Tugce Martagan is an Associate Professor at Eindhoven University of Technology. Coen Dirckx is a doctoral student at Eindhoven. And Annick Nusselder is a data scientist at Merck at MSD. I think I covered everybody over there. On that note, let me hand it over to the group. Take it away, guys. Thank you again, and looking forward to the presentation.
Hi all, my name is Oscar Repping. I'm a biotechnologist by study and professionally active in this field for over 22 years. And we are pleased to present to you in this M&SOM seminar today. Biotechnology will have a central focus in this session. Have you ever worked on a jigsaw puzzle and could not find that single piece to complete it? We are going to present to you where we are with our jigsaw puzzle at this moment. And our jigsaw puzzle contains only a limited few puzzle pieces at this moment. And most of these pieces do not exist yet. And it is not clear how this puzzle will look like when it is fully completed. Merck started a collaboration initiative with the Technical University of Eindhoven in the Netherlands. Our strive to make the unpredictable nature of biology more predictive was started around 5 years ago. And together with the Associate Professor Tugce Martagan, we are moving towards a predictable future in the biopharmaceutical industry. And if you want to learn more about my biomanufacturing and why this should become our new reality, you are at the right place. And we hope you will be as excited as us about the potential this could bring. First, we would like to bring you closer to our world, the world of biotechnology. And secondly, we will present to you our recent work and our vision towards operations research. And lastly, how does that future then look like? And we will end this session with a Q&A. So when I started to work for this company, the first thing they taught me was the belief that biological processes are by nature highly variable. So, and guess what? I believe them. And in time, I learned the tribal knowledge on how to set up and influence these processes. And these processes remained highly variable. And during my development within the field of biotechnology, I actually came to the conclusion that we should use our data differently than we ever did before. working towards a data-driven decision-making model instead of best guess on color of a culture. To date, competitive advantage in biomanufacturing has been primarily driven by the science, and in other words, our knowledge of the underlying biological and chemical processes and how to exploit them to produce active pharmaceutical ingredients. But as market demand and competition have increased, the industry is now more sensitive to cost and production footprints. We are under a continuous pressure to make our biomanufacturing operations smarter, reducing cost and lead times to create the critical medicines that we need. So transitioning from science labs to smart operations is not easy. It requires a new vision and a transformation towards smart operations that proactively uses data analytics and operations research to inform our daily decisions. The big question is, how can we leverage our data as effectively as possible to solve the many challenges we face in biomanufacturing? The industry would benefit from an interdisciplinary approach, working in close collaboration with the informed community to harness the power of cutting-edge operations research and operations management methods to complete our life sciences and biomanufacturing expertise. Our team is from Merck Animal Health, but I would like to highlight that our approach that we are sharing in this seminar is also applicable to human health. The manufacturing of biopharmaceuticals uses the same techniques, equipment, and operational challenges, whether the product is destined for humans or for animals. The animal health industry faces additional pressures that make it critical for us to improve our biomanufacturing efficiency. So most regulatory requirements between human and animal health are equally stringent. Market demand for animal health products is increasing. The animal health industry at large cannot currently produce enough supply to meet the ever-growing market demand worldwide. And most importantly, unlike human drugs that are often subsidized, animal owners are responsible for paying full price of these drugs. From a farmer's perspective, the price of a vaccine needs to be sufficiently offset by the value of or revenue obtained from a healthy animal. Within the animal health industry, we continuously need to be more creative to meet the needs of our customers. So let us move forward to show you how we move from tribal knowledge to operations research-driven decision-making.
Okay, so let me briefly review the current literature. Right now, when we enter the keyword biomanufacturing to Informs Journals database, there are only 9 research papers with this keyword in all Informs journals. When we switch to biopharmaceutical keywords, there are slightly more papers, but still not that many. So the good news is there's a big room here for contribution and impact. And at the same time, we also project that the number of papers will grow exponentially in the near future. This is because, first of all, we have new sensors, new technologies, and that way we are able to collect various types of data or control many different parameters in real time. This was not possible in the past, but it's now possible with these new technologies. In addition, as Oscar mentioned, the industry also recognizes the potential value of ORMS, and there's currently a clear shift in the industry's mindset from tribal knowledge to more data-driven, more OR-driven decision-making. And this is why we are here today. We hope to stimulate and inspire new research to help this field grow. Now I will hand over to Bram.
Hi, thank you. So let's first address the term biomanufacturing. What does it refer to? Well, biomanufacturing itself is not a it can be defined of any process in which we use biological processes or biological systems to produce a component or a product. So looking at it that way, we can conclude that biomanufacturing is with us already for centuries, because in the classical approach, making yogurt, beer, bread, or cheese can be considered biomanufacturing. Looking at to it from a more modern perspective, the manufacturing of medicines like antibiotics or hormones like insulin, vaccines, or even immunotherapeutics for cancer treatments, that is a really good example of modern biotechnology. So to characterize a bit the process is these components are generally produced using batch fermentation. Well, what does that mean? It means that we use a single bioreactor, and that bioreactor is depicted in the right top corner. A single bioreactor, and we start the fermentation with all components in place. We let the fermentation proceed, and at the end of the fermentation, we harvest the full batch. When a batch is finalized, in between there's a full changeover, which means that we have to clean and sterilize the complete bioreactor, and we can start over the new process. So in the middle picture on the right, you can see a continuous timeline of such a bioreactor where you have the setup time. You can actually produce a culture, and then after the harvest, we do a new setup. So you really see a flow here. So looking at a little bit at the biology, the underlying biology, when a microorganism grows, we can divide the full process into 4 stages. When we start a culture, it starts with a lag phase, and the lag phase really means that the microorganism gets to adapt to the environment. And after the lag phase, the culture transitions to the exponential growth phase, which means that we have a maximum accumulation of biomass in the system. The microorganism starts growing, it starts consuming nutrients, and at some point the nutrients get limited and the culture transitions to the stationary phase. And in the stationary phase, the growth rate equals the death rate. And this transition phase, from a business point of view, is a very interesting transition, as the amount of product made per time unit starts shifting, and actually stationary phase is even a phase that does not provide enough business value anymore. So interesting about these 3 phases is that it follows an S-curve, which mathematically seen it can be well described. So moving on to the next slide. If you would review the concepts of biomanufacturing, what I just presented is the batch manufacturing. And the batch manufacturing is depicted on the left side, and is the most commonly used. And if you would describe the S-curve in time, biomass versus time, the picture is below the batch culture. So other concepts of biomanufacturing that are described in literature and also applied in practice is, for example, the bleed feed, which is a mechanism where you have you start a culture with all components in place, you start growing, and at the transition state, you partially harvest your culture, let's say 90%. The remaining 10% serve as a Kickstarter for growth for a second culture. So you replenish with fresh nutrients, and you actually have a second growth curve. The value add of this is that you actually skip your setup phase, which reduces time and cost. A more complex way of biomanufacturing is perfusion or continuous stirred tank reactors, which is more of a continuous process where either you have a recycle of your product and you have an outflow of waste, and because of an outflow of waste, you have a room to feed nutrients in, and in this way you can extend growth phase. Or the other way around is that you have an outflow of product, a continuous outflow of product, which allows for a continuous feed of new nutrients. And also this enables extension of the growth phase. So going to the next slide. As already introduced last year during the Edelman presentation, we were able to show that we're able to combine operations research and biomanufacturing. And we've shown that we're able to effectively and efficiently implement operations research in biomanufacturing, that we were able to scale up the processes, but also to transfer in between factories the applications. With those examples, we have proven a significant impact, but also we've shown that there's a huge potential to transform biomanufacturing at large. During this seminar, we are going to focus on 3 research directions. One of the research directions is smart biomanufacturing. We will follow that up with real-time release, and we'll finalize with the agile network supply. Over to Coen.
Yeah, thank you, Bram. So the first topic we want to tell you something about is the data-driven decision-making in biomanufacturing, because in practice, we often encounter 2 cases. Either we have a very limited and small data set, or we have a very large data set. And both these situations, they have their own unique characteristics and also their own unique challenges. So for the small data set, which we often encounter, you have a limited amount of historical data. And this could be due to several causes. For example, maybe it's a new product, or it's even still in the R&D phase. So then by definition, you only have a limited amount of data. But we also see it in our large-scale production facilities. For example, once we change something in the process layout, or we start to use different materials, then you need to start again with building your complete new historic backlog of data. So we often encounter that we have only a small amount of data to work with. Also, next to this, in terms of sensor technologies, we also encounter some limitations, which make data collection quite challenging. For example, several process parameters we monitor in line. Think of temperature, think of pH, and we have to do that to make sure that this process is within its operational boundaries and doesn't deviate from it, and where needed, such that we can steer it into the right direction. But of course, there are many other parameters, and you simply cannot monitor everything. Well, in theory you could, but there's always this trade-off. Is it worthwhile? And some of these critical process parameters, ideally, you would like to measure them in line. Think of a biomass concentration, think of the number of cells or the number of infected cells. But in practice, it's common use to measure them in an offline fashion, just enough so that we sample every few hours to measure the batch quality and to make sure it remains safe. But these tests, they are quite labor intensive, time consuming. And by definition, this kind of approach makes it hard to use this info for real-time decision-making. So this leads us to our first research question. How can we make smart decisions in the presence of limited data? And as already pointed out, with the advance of new sensor technologies, in the future, which data should we start to collect? And how do we make best use of it? Well, to address these kind of questions, future research could focus on the development of new methodologies or decision support applications, for example, using Bayesian learning or black box kind of models. Another thing we often encounter, since we're working with living organisms, is variability. And this is actually a critical challenge in biomanufacturing. Since we're working with these living organisms, there's a high level of batch-to-batch uncertainty. And as a result of this, a fermentation process is inherently quite unpredictable. For example, the figure you see here at the top right corner, it plots the cell growth rate of a product for different batches. And you kind of see some small deviations in it. But the funny thing is, we actually did exactly the same steps for each of these batches because we have to follow a standard operating procedure. But still, as you can see, the outcome is slightly different, even like in the growth curve pattern, but also in terms of the quality, whether it's a good batch or because the batch was bad and had to be rejected. We also encounter such batch-to-batch variability, even in the raw materials being used and the reagents we add during the process. And this all contributes to this uncertainty and bioreactor outcomes. So not only are these processes highly variable, it's also combined sometimes with small data. And this is definitely an interesting challenge for building good prediction models. On the other end of the spectrum, when we do have sufficiently large data sets, our research focus really changes. We start to focus on how can we develop accurate and explainable prediction models. However, a prediction model for the evolution of a biological system, that's not that straightforward, since they are highly complex, non-linear, and also often non-stationary. And in the life science literature, there's considerable research, mostly on so-called kinetic models, trying to explain, okay, how does these microorganisms behave in a reactor? But the problem with these kind of models is they are almost all deterministic. And as we just showed, there's much variability in these kind of processes. So these observations, they lead to several research directions. For example, how can we develop new models and methodologies to link these variable cell level dynamics with the operational trade-offs and the business risks? So on the one hand, you have these differential equations and stuff like deterministic modeling, which we would like to combine it with our knowledge from operations research to incorporate the stochastic trade-offs. And in addition to that, these kind of models, they should be explainable to bioreactor operators, as well as to regulatory agencies. And therefore, we foresee a significant research opportunity on hybrid modeling using AI-driven methodologies and reverse model design. Next to these optimal control algorithms I just talked about, we also identified several other practical relevant research directions. For example, new approaches and tools to support new technology adoption decisions. For example, what is the business case for perfusion? And when is it beneficial to adopt these kind of new systems? And also looking at different product strategies. For example, here it mentions subs versus stainless steel. Subs is a single-use bioreactor. So it's basically a reactor you only use once and then you have to throw it away. For example, a plastic bag. Versus a stainless steel reactor, which you can use multiple times, but there are more cleaning costs involved. Other decisions you can think of, should you produce in batch, in bleed feed, fed batch, or even in perfusion, which is a fully continuous system. Next to that, you need to think of what is the right level of automation for specific products or biomanufacturing processes. Because for some processes, it makes sense to collect more data, but that is also more expensive. So there are some trade-offs to be made there. And lastly, it is good to, of course, start optimizing your production environment, but you should definitely not forget that you have to take into account your upstream and downstream processes, because otherwise, yeah, simply your bottleneck starts to shift. And therefore, it's really important for system designs, take factory dynamics into account, link your fermentation environment to planning, scheduling modules, and really capture that interaction as well, more on a higher level, let's say. Yeah, as explained earlier, most research in life sciences really focuses on these deterministic models or empirical analysis to understand the biology and the chemistry behind these processes. We actually foresee that stochastic modeling and optimal control of biomanufacturing systems can generate a substantial impact on the industry. Although several other industries have already benefited from operations and management science methodologies, there's still so much to do here in biomanufacturing. It's still actually, as Tugce already showed, a little bit understudied. So we like to see that as a real opportunity. Well, to illustrate the potential applications of operations research methodologies in biomanufacturing, we provided 2 examples, which Bram also already talked about briefly, the bleed feed and the perfusions. And in both examples, we have really the central research question, how to optimally control these highly non-linear and uncertain systems in real time. And in particular, how to capture the high complexity of these kind of systems, model it, but do it in an elegant and easy to implement and still a realistic model. Well, for the bleed feed, you really have this dual trade-off because on the one hand, you don't want to bleed too early because then you could have gotten more out of your batch. But on the other hand, you don't want to do it too late because then you may miss your window of opportunity. Next to that, there is another decision to be made. How much are you going to bleed? So you really get this kind of dual trade-off. And on top of that, you have to do it under uncertainty because each batch is slightly different. Well, our forthcoming paper in the M&SOM presents in the M&SOM journal will present the first attempt in modeling and optimization of bleed-free decisions, but there's still a significant research opportunity even in this direction. And then perfusion systems, which is really an exciting direction for future research, because this is by definition, this is continuous manufacturing. Well, perfusion and continuous biomanufacturing in general, they are relatively new technologies, and they should also be more explored. In this context, we are really in need of new models, methodologies to optimally control perfusion systems in real time. And in order to do so, we would like to first identify a golden profile or a target trajectory for the evaluation of the fermentation dynamics over time. In order to do so, we need to harness our expertise not only in life sciences, but combine this with, for example, AI-driven methodologies to identify this golden profile, and when it deviates from that, steer it back into the right direction. In addition, we also need to optimize several other operational parameters, such as the feeding rate, recycle rate, harvest rate, the duration of the campaign, and some harvest decisions. And with that being said, I would like to introduce our next challenge.
Our next challenge is the real-time release testing. And before I explain what our research implies with real-time release testing, it's interesting to know what real-time release testing is. So real-time release testing is a novel control strategy where, as we are currently producing our products, we are measuring the quality of our output. So you're taking samples and we physically test the product. With real-time release, we want to move away from this testing of the product, and we want to predict the outcome based on how the process was being done. So as Coen already mentioned, our process is stable, but we do have process parameters that can indicate certain relationships to the output of the product. So three major directions within real-time release testing. The first one being prediction models. So as I said, with real-time release testing, our goal is to move away from testing our product, but predicting the outcome. And for this, we need prediction models. If we look at how our process goes, we have an ideal process flow. If we look at, for example, of the growth of the organism, and this is often captured in a golden profile where we map the process in a certain growth pattern. And by that, we can predict the outcome, what we have in the end of the process. As we produce batches, we want to dynamically learn and update this prediction model and update that golden profile and better predict the process. So our second challenge is process modeling, process optimization. If we look at real-time release testing, we want to have a better look at the process in general. We need to model it and we want to optimize it. It also gives us the opportunity to improve other things of our process. So as soon as we start to predict the outcome of our process, we can see if certain aspects of the process may not be as we would expect or in normal way of occurrence. And by that we can, for example, see if we would need a certain maintenance, and instead of waiting before the maintenance is actually needed, we can go to preventive maintenance. Also, we need to know when we can skip certain inspections. So as I mentioned, how the process normally goes is that we take a sample and we physically inspect it. And there is two ways of using real-time release testing. We can always skip inspection, or we can say, let's predict what the outcome will be and decide when we should inspect the product. And when we have a reliable result, we can skip this inspection. Then as last, factor dynamics. So if we look at a factory, it does exist of multiple production processes. So we start with raw materials, then we produce an intermediate product, and in the end, we have our final product with some in-between steps. And if we want to do prediction modeling and predict our outcome, we want to take this information through the whole chain of those processes. And we want to create traceability. And also very important is, when do we say that a product is seen as a good product or a bad product, and when do we trust our results? For that, we need to have the business implications and the risks, both in monetary, but also in quality of false positives and false negatives. So what happens if we do have a result that is not reliable and would differ if we would have taken a physical test? And these are all the challenges that we have at real-time release testing, as it is such a novel way of controlling the process. There is not much research there. So I think there are great opportunities here to have operational research help us with this challenge.
Now let's look into agile network supply. Biomanufacturing facilities are highly complex, while one manufacturing division consists up to 10,000 interdependent process steps. The same product can be produced with different process steps or alternative production paths. Some products have preferred paths, and not all paths have the same yield. Pushing a biomanufacturing facility to its capacity limits and/or expanding capacity generates several complex questions that must be answered before successfully delivering a capacity increase. Often, plants don't have a clear and obvious bottleneck, and the capacity may be limited by a series of steps across many pieces of equipment. This is where OR can make the difference. In biomanufacturing, the challenge with scaling up is that the upstream process of scaling up changes the cell culture microenvironment, thus impacting product quality and process characteristics. In contrast, scale out eliminates scaling up because the bioreactor size stays the same throughout clinical and commercial manufacturing. Increasing scale is thus achieved by increasing the number of bioreactors. In scale up, an unexpected loss of a bioreactor would create huge financial and time losses. However, with scale out, you would lose just one of the several bioreactors used in a production run. But this approach comes with a price, as the running cost will be higher. Data-driven models can give insight on the trade-offs to make the right strategic decisions over time. And it would seem intuitive that biomanufacturing cold chain distribution would lend itself to a lean, just-in-time inventory model, because medicines and biologics are expensive to produce and can be stored for extended periods. COVID-19 has demonstrated the risks to that model, from supply chain uncertainty. At the same time, the perishable nature of drugs and the loss of efficacy over time jeopardizes resiliency strategies dependent on safety stocks. Finding the right balance to optimize inventory based on the product portfolio, historic demand, and shipment patterns will be a continuing challenge. OR-based simulation and optimization models are critical to ensure accessible and affordable medicine over time. During this presentation, we have tried to give you an insight in our journey so far and the puzzle pieces we have collected along the way. But as you can see, the puzzle is far from complete, and there is a vast number of opportunities to explore within the intersection of OR and biomanufacturing. We hope that we inspired and stimulated you to join our journey towards more operational research in this field. To get you started, we will follow up this seminar with a research paper, where we will elaborate on the details of our research roadmap. In that research paper, we will also provide real-world data to the Informs community to support and inspire the development of new methodologies and decision support applications in biomanufacturing. Together, we can make this world a better place for all.
Thank you, Marc. Thank you very much for your attention and the questions that we have seen already in the chat. Hope it made sense, the presentation that we gave. We are, of course, open for questions that we will try to answer as good as we can. So we can get questions via the chat, but feel free to unmute yourself and ask the question that you have on top of mind.
Wonderful, and I'll just start it off but if anybody has a question, feel free to unmute yourself or raise your hand or chat it in or however you want to do it. But I was curious what the implementation challenges were, because I'm thinking about other contexts where similar transitions have happened. You know, retail, but if you go back many, many years, the world of finance, you know, where it used to be a very tribal knowledge, you know, but the investment management I'm talking about, you know, and that the transition there has been much more dramatic. But in every case, there were significant implementation challenges. So more familiar with retail myself, but even in the financial world, you know, worlds don't change without wars. We've realized, right? It's so just what were the challenges that you have encountered so far in making it work in practice? You know, it's one thing to come up with how to do it, and well, anyway, I can see from your body language you seem to understand the question. You can even amplify what exactly that means.
Yeah, and Andra, that's a fair question. What journey are we walking? Which is not only had a pure operations research science, but besides that, even if we would have, let's say, a perfect model or a perfect tool, one of the most critical elements is the acceptance of the people to use it. So it's our biotechnologists, our biologists with their tribal way of working do not accept that easily, new ways of working. And so that's already one. So the change management process in this is instrumental. And we actually focus in three high-level buckets, which are very simple. We talk about people, processes, and tools. And what we are doing here is creating tools. And the element that we never should forget. A tool without a process, a tool without capable people is a tool that is not being used. Mm-hmm. So that is where we are. Besides exploring operations research and implementation, that is how from the other angle we are looking to this entire program, because it's much more than the pure science. So if between those two extremes, we are progressing forward as such, with all complexity that there might be. Does that answer your question?
Answers very well, but maybe others might want to augment in some way. But yes, it does answer my question very well.
Maybe Anand, I can try to augment here. So there's not only the people inside the company, but there's also obviously the regulatory authorities that are, you know, closely interwoven with this. So my question is then, you know, especially like this one of the topics you brought up was this real-time release testing and skipping tests, right? So that would obviously require the authorities to buy into this. So what challenges do you see here?
Yes, and that's a great question, Martin. And let me ask Annick to explain that, as she did have had contact with our regulatory department to find out what do we need to what risk do we need to materialize, or what should we prepare to move forward to authorities. So Annick, if you could answer this question, please.
Yes, definitely. So with real-time release, one of the main challenges, or actually there are two main challenges. One thing is the explainability of the model. So what is always the trade-off is how accurate the model should be, that is incorporating many different parameters, many different aspects within the modeling. But also for a regulatory perspective, it needs to be very interpretable. They need to understand it to be able to trust that approach of testing and to have it approved. So that's for sure the biggest challenge that we have. A second one is that the data is often quite limited. So we don't have so many batches. So how do you get enough information, even though with regulatories, they have some kind of information on what criteria you need to meet. But as it is such a novel control strategy, there are no biomanufacturing companies that have actually be able to get it approved. So we don't know how the perfect looks like. So I think really here we need to have that communication with the regulatory parties.
I see that, Nicola, you posed a question on chat and Tugce actually partially answered, but maybe you can augment the question for all of us, provide some context, and because, and then maybe the group, including Tugce, can respond more.
Yes, I just saw that. Thank you for prompting me to elaborate. I just saw the answer, which is great. I don't have much context because I don't know biomanufacturing. I was just thinking what that community is doing. So the engineering community, in particular chemical engineering, what they are doing, and whether there is a way to take what they have done or are doing and integrating it with what we do or what this project has been doing, and support each other. That is what I was thinking, but I don't know details. That is why I was asking.
I think you make a very good point there. I think there's so much overlap, and indeed it starts at the basis with chemical engineering, and currently they're really focused on these deterministic models, which for a chemical process that is often true, right? Under the same temperature, under the same pressure, product A and B become C. But here, if we do that 100 times, then we get 100 times a different outcome, but still the process and basics is the same. So if you can combine that with our knowledge on how to deal with these stochastic situations, that's really where you can harness the synergy. Definitely a good point.
Good, thank you. And I was also thinking about bioenergy. There's got to be some way to bring what you've done to bear on that domain.
And I suppose there are two, this might be a very naive question building on what Nicola just said, but I guess there's two ways to go about it. One is you can actually, Coen, for a young doctoral student like you, is reduce the uncertainty, right? Through, I guess, what you call better prediction models, if I'm getting the concept right, The other is, okay, we accept it and we do the best stochastic optimization that's possible. And it's not one, they're not mutually exclusive, but both seem like very attractive paths to pursue. And just going back, sometimes optimization becomes harder when there's less uncertainty, right? I mean, this is at the extreme. If you know nothing, anything is optimal, right? But as you gain more knowledge, the optimization challenge can become more significant.
And maybe to add on that, from a manufacturing perspective, if we're able to better predict using the models to predict a certain outcome, in other words stated, to reduce the variability, that helps on so many levels towards our customer, because you'll get a better and more stable quality in your medicine supply. That's for one. But the other one as well is linking to the real-time release testing. In order to get the authorities to get along with this and to actually understand what we're doing, the better we are in control with our variations, the better we are in control with predicting what parameter influences what outcome, the better we're able to sell the whole concept of real-time release testing. And I think this is one of the biggest challenges at this point, because looking at authorities, they're really open to the whole concept of real-time release testing. And it is in the regulations already that you're allowed to do so. Looking at particularly the small molecule pharmaceutical industry, some real-time release testing is already in place. So at small portions, this is already being accepted. But the harder the mathematics get, the harder the more increase in parameters we get, the more difficult it is to really take the authorities along in this journey.
Yeah.
how are your competitors approaching this domain? And that could be, it's mixed news. Well, go ahead. And then, because sometimes it can actually be great news if they're also doing the same thing.
Yeah, and I am not completely sure, but Tugce, you explored what publications are out there. You have had contact with other companies. Maybe you could help to answer this question from your perspective.
Yeah, so I understand that the real-time release is actually successfully used in the world of small molecules. So when we have, for example, more like deterministic processes, but when it comes to large molecules like we are discussing right now, it's still a dream for the future, and I know that people are actively working on this direction. I know that this is a relevant topic for the competitors as well, but we still need to make a lot of progress. And in addition to the things that we discussed right now, there is also an ethical aspect of this discussion, which could also be relevant for the informed community. For example, in the future, if we skip those tests, and something happens in the outcome and we do not catch it, then it has huge implications for the whole society. So when we let AI make decisions, then who's responsible? So these are all different types of discussions which are relevant, but I think it's good to make all these discussions to help this field grow.
Okay, so here's at least I said if many people on this call have never worked in this domain, right? But if they wanted to, if any of us wanted to get into this domain, how would you suggest we start? I mean, sure, we can pay attention to what you're doing, but go ahead. I'll let you take that question. How would you? I know there's somebody you guiding Coen on how to do it, but what about others?
Yeah, that's. I actually like that question. This is also one of the reasons that why we are presenting here in this M&SOM seminar. We would like to find out: is there more interest? Are there more universities interested to join this fundamental research? And how could we make sure? And we hope that Tugce would like to organize and coordinate this. How could we have the study plan that we now have? How could we make sure that we can enhance, expand, enlarge, accelerate the research? Mm-hmm. We need more brain power. To do that, and just as an FYI, we have limited opportunity to measure inline our processes. Yes, the normal well-known parameters like dissolved oxygen, like pH, like temperature, like biomass, those are relatively easy to measure, and we know how to interpret and analyze the data. There is also another type of technology, that is the infrared NIR technology, which we would love to use in our processes. So if we start to do that, and if we, for instance, start to implement Raman technology into our processes, we need to find out what are the how to interpret the data and how to find the correlation between a parameter that we are interested in into the data. This is an immense complex exercise, and this is definitely something that we are interested in, and to find universities that would love to collaborate with us in the near future. But I love the question.
Yeah, maybe to add there another, because I think it's a fair question, right? Maybe one of the reasons that this is kind of An unknown for many is, of course, that there was not too much publicated from different organizations in this field, right? So we really said to ourselves, if we want to lead the way and really make this place make medicines affordable and more accessible, we need to be a pioneer where we kind of reach a hand to the community and say, hey, if you want to join on this journey, this is the way to join in. And that's also why we will follow up this seminar with not only a research paper with our future direction, but also real life data, right? Where everyone in the community can start to explore and get excited. And from that, we really hope that we start to get a kind of a new research area opened up that maybe even the whole within the community, other companies are joining in on. But in this case, it's a fair question. And we hope that by publicating and presenting the way we currently are doing will help to open up opportunities.
I'd love to hear from people like Georgia, Jeremy, Pavitra. I mean, there's a whole bunch of people over here who, Stephen, who've been doing research for a while. How do you see this gaining traction? And I think one key word over here, I would say, Tugce, is persistence. Don't give up, you know. But grit and tenacity go a long way in any field, all right? But okay, and evidently Georgia is saying yes, or at least saying thumbs up over there. But how it seems like a really interesting domain. We sometimes, in certain domains, what we've done is gone in and then explained what practice has discovered. And that's a useful contribution. I think what I'm hearing from the group is we can be part, or Tugce is already part of the process of making that change, you know, which is an exciting domain to be in. But you know, we'll also have to train students, and not just doctoral students, undergrads, master's level students, because the team will need to hire not 3 people, but 300 or 3,000 people, you know. It's so it's got implications in all domains. But how do you guys see this playing out at your schools, for example?
Maybe I can start that. I'm currently part of a group that is AI for Industry 4.0. So here, I think when I hear about your problems, I see a very similar kind of discussion happening with several manufacturing clients who are not in bio, but they want to use AI for optimizing their manufacturing process, and they are getting similar temperature and other kind of sensor controls. They're developing AI models. They want to help the controllers with automation to improve the yield in these different processes. So we have also worked with, say, food manufacturing companies. We have worked with automotive companies, and the theme here I see is sort of very similar, that you have these very complex black box systems which you are trying to have AI models to predict what is happening, and then you want to have some decision-making possibility to control various aspects. It could be all the way from yield to sustainability, and then you have these chains of manufacturing process to not just one, but you have groups of them, one giving inputs to the next, and how do you optimize this entire chain? So I do see a lot of parallel in what you are saying and what is being done, and so maybe the idea is to get this more larger theme that is there, that we can actually pull together across industries. Yeah.
So is the larger theme, Pavitra, as you see it, industrials or process manufacturing? Because there are many ways to slice it. And in fact, Tugce, viewing the way Pavitra was looking at it, maybe the we are different from traditional manufacturing in that context might not be the way to frame it, you know, in fact, the theme might be we are similar, guys. You know, we're all changing, and we're, of course, the flavors are a little different, you know, because maybe that's the group. But Pavitra, how do you see the necklace forming over here?
I mean, again, like I have a feel for pricing, so I always connect it back to pricing, but it is sort of, you're trying to model demand as a function of price. So you have this behavioral thing. Here you're trying to model this black box system as a function of various control parameters. And then you're going to go as a next step and put these complex black box AI models, which are not like your linear or anything, right? There maybe you might use like your fancy deep learning models or something that goes inside your optimization, which then you have other objectives for, and you're trying to optimize following that thing with various goals. So I see this, and then you have, I mean, again, that is more like if you think of it as one thing, but then you have like a sequential decision making, because these things have downstream impact. You have to consider their time indexed formulations, their network formulations. So I see that we in our community have a lot of these tools which we can actually adopt to a variety of these different settings. And I think, I mean, that's how I see it coming together. Yeah.
And since we don't have that much time, I want to make sure Jeremy and Steve Cheek, who is also on the call, speak, since I would love to hear from them rather than me doing the talking.
Actually, I've got a really quick one. I'd love to hear from Steve as well. But, you know, it strikes me that, you know, an academic in this call with a lot of experience doing really good academic work with the biomanufacturing partnership with a biomanufacturing firm is Tugce. And so I was curious, you know, if I can put you on the spot, Tugce, if you had any insights or thoughts on what it takes to do good academic work like you have in collaboration with the biomanufacturing industry, which seems very specific, maybe for the depth of the technical aspect of knowledge in addition to all of the operations problem. Could you share a bit more about your experience of doing practice-based research work with biomanufacturing experts like the other speakers on the call?
Well, thank you very much. I think that's a very nice question. So if I look back to my experience, I think the biggest part was the beginning, because I have no training in chemical engineering or biological engineering. So when I tried to understand what was happening, it was really difficult for me. I still remember as a PhD student, sometimes I was trying to interact with professors from chemical engineering, but I was not able to follow up the discussions because it was too technical. But after a point, when you start to catch up with the biology and chemistry of these processes, then as we just discussed, the rest, like building the AI models, building the optimization models. So these are really, easier in my opinion. So breaking the barrier, understanding the processes, in my opinion, as an industrial engineer, is the challenge here. But that's the reason why we are also here today. We just want to educate people more. We will continue to publish more, especially as a follow-up to this seminar. And we will make sure that we provide some easy materials for the ORMS community, just like a pill, you know, Biomanufacturing 101, so that you can easily grasp that knowledge and then continue with the relevant ORMS methodologies.
Thank you very much.
And if I may add to that, the counter side of that, the explanation of Tugce from an operations point of view, I've had the opportunity in the past 5 years to guide about 10 students from the university in Eindhoven, which all did not have any biological background, but really were operation research focused. And it's really good to see that apparently the biological concepts that really matter to make this first step into applying operations research, it's a step in knowledge that can be taken, I think, within a month. Within a month, all of the students were up and running, grasping at least the concept of the biology, the things that were going on. And of course, if you want to go more in depth, then it takes a lot more time, of course. But I think just showing that within a month, at least 10 cases that I've seen were able to get along, to go along, and to show that they can create impact, that really shows the easy connection that we can make with several backgrounds to merge into this OR biomanufacturing principle.
Okay, I think we are actually out of time, but Stephen, do you have a 30-second or, you know, before we sign off? Because Georgia cold called you, right?
Yeah, sure. Yeah, I even turned on my video so you could see my beautiful hat. I'm super happy to see this this webinar. Thank you so much for doing this and for organizing it. I was so glad as well to hear that the you know, Biomanufacturing 101 primer would be around. That's super important. You know, and because all of these technologies are very different. You know, I first got into vaccines by talking to people who, you know, were running a vaccine plant, spending time with people working in the plant, you know, and just reading these basic primer books. You know, this is the book that I used to kind of come up to speed with the vaccines on. And having that contextual knowledge is so important of what's relevant, what's not relevant. So that primer is super useful. And but the biological production, which we didn't talk about, you know, the different flavors, having a sense of, you know, what's different between the monoclonal antibodies, what's going on with the various cell therapies, what's going on with, you know, in this case, the egg, you know, working with the eggs, just having a flavor of some sort of a taxonomy. This would be really great. And if as a community, we were able to share and pool and refer, I think that would be a really useful thing for our community, because there's so much going on. And it's going on not just in the US, right? It's going on in a lot of places, and there's some really neat things going around, you know, reducing cost of production. I think that would be really fun to see and understand, well, what's driving these and how can we help support some of the new possibilities with this? Thank you so much for doing this.
Yes, in addition to thanking, I just want to say maybe a thing for the rest of us, all of us who are in academia, to think about is whether there should be a primer or a primer series, you know, as part of our journals, not necessarily a talk to say what are future research directions, but hey, here's, you know, Biomanufacturing 101, all right? It's so people know that, or even if they don't have a way to reach out to Tugce or somebody like that, where can they get up to speed? So we are out of time. Michelle's been messaging me and saying, you know, but I would like to reiterate our sincere thanks from everybody over here. The video will be available. Tell your friends. I think it's an excellent call to arms from our group over here. So thank you very much.
Welcome.
Okay.
Yes, thank you for having us.
Thank you.
Yeah, thank you very much.
I guess.
Bye-bye.
Okay, see you.
Happy
Bye-bye.
Friday.